Hello, everyone, and welcome to session number 76 of the Porous Time Talks. I am Chiara, and I'm joined here in the studio today by Sarah and Marcel. And before starting the official session, we are very excited today to have a little announcement. So Porous Time Talks is growing, and two new members have just joined our organizing team. We have Rima Manik and Berilo Santos. Rima is a PhD candidate from the Indian Institute of Technology, Hyderabad, in India, while Berilo is a PhD from the Universidade Federal de Juiz de Fora in Brazil. So welcome to the team, Rima and Berilo. We are really happy to have you here with us. So as usual, with this platform, we aim at giving the stage to early career researchers to present their work to the wide world of porous media. And today we will have two great talks. I remind all the people that are connected online on YouTube that you can post your questions and comments, and then we will read them out loud during the Q&A sessions after the talks. So our first speaker today is my dear friend, Wenqiao Jiao. Wenqiao holds a double PhD from the University of Lausanne in Switzerland and from Politecnico di Milano in Italy. He has just been awarded, actually, an SNF Postdoctoral Mobility Fellowship, and he is currently a postdoctoral researcher at the Department of Civil and Environmental Engineering at MIT in Boston, where he focuses on the mechanisms of plant root growth in porous media. Today, he will not talk about plant growth, actually, but he will present a part of the work he did during his PhD between Lausanne and Milano. And specifically, he will talk about the dynamic changes of heterogeneity of porous media architectures that are induced by the growth of biofilm and how these impact on the overall permeability of the system and how we can actually predict that. So with this, Wenqiao, the floor is yours.
Yeah, thank you for your introduction and the invitation. I'm very happy to be here to share my work. And I'm Wenqiao, you can call me Leo. Today I will...
Sorry, Leo, you should...
Yes. Today I will show you my PhD work between University of Lausanne and the Politecnico di Milano. My research focuses on the structural heterogeneity and the permeability of the porous media. As we all know, complex porous media host complex flows organized in preferential flow paths and stagnation zones. This impacts the transport and mixing of solutes and colloids, as well as their filtration on the solid green walls. Also, bacterial transport through the porous media depends on the structural properties, leading biofilm growth in complex architectures and microbial-driven bioreactions, such as biomineralization. So accurate understanding how the structure controls the flow and transport through the porous media is very important for a lot of applications, such as real bank filtration, deep geosome energy production, and CO2 storage. From a macroscopic view, Darcy's law is a very powerful tool to relate average velocity and hydraulic gradient, but it does not help to understand the relationship between the overpermeability and the macrostructure. For homogeneous geometry, from a microscopic view, taking into account the detailed friction experienced by the flowing fluid on the solid green walls leads to well-known Kozenik-Hammar formula. It predicts well the overpermeability to scale as the average green size squared and the global term, porosity term, like that. But still, it does not help to predict the overpermeability of heterogeneous porous media based on their macroscopic structure. However, in realistic world, the porous systems always have a heterogeneous structure with a wide distribution of per size and per shapes. Moreover, the structure can be altered over a short time by a lot of phenomena, such as biofilm growth and mineral dissolution. The biofilm growth reduces the size of the local pore, and the mineral dissolution have opposite effect. So today, let's discuss about how the heterogeneous structure control permeability of the porous system and how the heterogeneous structures control the transport-driven processes in the porous medium. Let's focus the first case, intrinsic permeability of the heterogeneous porous medium. First of all, I designed 12 heterogeneous geometries with different grains organizations. The green circles represent the impermeable solid grains. Then I characterize these geometries by defining a pore in terms of a donate triangulation, where each triangle represents a pore. A pore throat lambda is the length of each edge minus the sum of the green radii, and the pore body is the distance between two triangle centers. From a global view, we can find the average grain size, porosity, and the pore throat, even the pore size, are very similar among these 12 geometries. However, by design, the distribution of these quantities is very wide. For example, the pore throat size can span across more than one order of each geometry. So how the spatial variability will affect the overall permeability? To investigate that, I fabricate the transparent porous media by printing the design geometries, and I developed an experimental setup to measure the permeability of each device by imposing a known pressure drop and measuring the flow rate at the outlet based on a balance. Based on Darcy's law, I can calculate the experimental permeability, but to do that, we have to consider the pressure loss in the tubing and the pressure loss in the inlet and outlet grain-free parts of the chip. We can calculate them by the Hagen-Poiseuille law. The pressure loss represents the delta PL here. For the pipe, we can calculate the pressure loss in the tubing by considering the cross-section is a circle, like delta PL1. For the pressure loss in the grain-free parts of the chip, we can consider the cross-section is a rectangular. You can see the equation of the delta PL2 here. Also, we have to consider about the volume variation of the inlet and outlet reservoirs. Then we can find the measured values are not predicted at all by the Kozenic-Hammer formula. We can see it from the right figure. That means the medium heterogeneity must be taken into account. To do that, I built a new model by considering the whole porous medium as a collection of smaller porous media in series. The overall pressure drop is given by the simple of sum of the pressure drop across each medium, and the pressure drop itself can be calculated based on the local size and the permeability of the porous medium. So that the overall permeability is the harmonic mean of all individual permeabilities, weighted by their own size. I propose here to connect the pore throat and the pore body to the medium structure. And the smallest porous medium I can conceive is a pore. Each pore has a non-hydraulic properties well predicted by the Hagen-Poiseuille law. Then, knowing the distribution of all pores, I can predict the medium permeability as a harmonic average of M pipe of permeability, lambda squared divided 32, and the length of W. The model predicts well the measured values, and as it takes into account the medium heterogeneity. To show the impact of that model, I redesigned 12 new heterogeneous geometries with the same porosity, average grain size, and average pore size. Then we can find here, cosine common formula always gives the same permeability. But if these average quantities are associated to their different per site distribution, my model would end with very different permeabilities as shown in the right figure. This work is published on the PRF. Now let's talk the second case. I will show you how the biofilm controls intrinsic permeability of a complex porous system. First of all, I design a heterogeneous porous media structure, and I characterize with the second method, maximum encrypt circle algorithm. Each blue circle in the figure E represents a pore. I want to study the impacts of the biofilm growth on the permeability of such geometry. So I designed a new experimental setup to grow a biofilm only inside the porous part of the chip by adding two UV light source on the top of the inlet and outlet of the chip to kill every cell eventually presenting there. I infected the chip with a suspension of the Puptida, a very common bacterial strain in the soil. Then I continuously inject a sterile nutrient solution by imposing a constant pressure drop across the chip with a pressure controlling system. While the biofilm grows, a result of cell division, more and more pores are occupied by biofilm, and less space are available for fluid to flow. Actually, I did two separate experiments with two different strains. One is the white type with flagella. You can see the right figure. A multi-strain that can swim within the porous medium. The other is a mutation, a normatized strain, and I repeated each experiment 3 times. As you can see in the figures, the line with dots is the average value. From a global view, we can find the growth of both populations seems very similar. The overall biomass agrees well with the classical bacterial growth model, the logistic model. However, effect of the normal type biofilm leads to the decrease in permeability to 6% of its initial value, while the multi-biofilm decreased to only 22% of it. With a very similar over biofilm growth. I agree that this is a result of the special organization of the biofilm and not its average value. So let's look at the detailed biofilm special organization inside of the chip. For the first 20 hours of both populations grew very similar, occupying the pore space almost homogeneously. But at a later time, the multi-strain does not occupy the second half of the chip. We argue that this is due to the depletion of the nutrients and microbial motility, allowing the cell to escape to an area with a high concentration of nutrient and oxygen, and this affects the macroscopic permeability. Moreover, we take the biofilm into account and assesses the permeability of the pore system. I extend the model I presented previously by considering that each pore, or we can say each MAC, can contain a certain amount of the biofilm measured by the tissue, the density rho. You can see in the figure B, the density of the biofilm is calculated based on the total intensity of the signals in each MAC and the surface area of the MAC. And based on the arithmetic average, we can calculate each local pore with each we can calculate the permeability of each local pore based by considering the biofilm grows in a pipe just on the interface. We can calculate with the density of the biofilm. It depends on the intensity of the biofilm. Then we can model. Then we can calculate the overpermeability of the whole porous media with a harmonic average. So the home take message is the heterogeneity of the porous media structures play a very important role in determining their permeability. And when the structure altered by the biofilm, the permeability also changes accordingly. And the porous structure affects transport and transport-driven processes with which it is then coupled. And as Kurt said, now I'm a new postdoc in CEE of MIT to investigate how the plant roots navigate through the heterogeneous soil and alter the spatial temporal distribution of their pore size, and investigate how the hotspots of the root exudate secretion drive the reservoir microbial dynamics, and how the beneficial bacteria inhibit the pathogenic fungi as the root grow at a pore scale. From the video we can find the tip of the root grows from the water into the air, and the green dots is the bacteria. It's just a test inside of macrofluidics. That's all. Thank you.
Thank you very much, Wenqiao. Really, really nice talk and really interesting topic. So while we wait for some questions to come in the on YouTube, I think we can start with questions here in the studio. So we start with a couple of questions of my own, a couple of curiosities actually, and the first one is like, so you have the biofilm growing, right, in the two cases for the two strains that you have, and I was wondering, I mean, do you have also like clogging of some channels inside your porous system, or are all channels still flowing? Let's say all pores still flowing pores, or do you also clog some of those? And if yes, how does this impact on the overall system?
Yes. I need to say sorry. I didn't explain clearly. Now I share again. Actually, in this figure, the color from the yellow to the black is representing the biofilm. We can see the biofilm clogs the pores. Mm-hmm. And the green is the gray circle is the impermeable green, and the yellow is the biofilm. We can see they occupy the pore space, especially at the inlet. We can find they occupy the pores, and the color is near black. They totally clog the pores. Mm-hmm. But here we consider about the biofilm is still permeable. This is why in the in my model, we use the only fitting parameter is the biofilm, is the permeability for the pore space with the biofilm inside. We can see the symbol KBF.
Okay. So basically, even if some pores are clogged, as the biofilm is not totally impermeable, you still have, let's say, diffusion of nutrients through the film itself, right?
Yeah.
Okay.
Yeah.
Okay.
And also here we think the biofilm is just grow on the interface of the pipe.
Mm-hmm.
From the figure C, we can see the flow can be divided into two parts. One is a pipe is total empty. The flow can be inside. The second is the interface. That means with the biofilm inside, the flow still can go through, but it's a kind of porous medium.
Okay, thank you. Do we have other questions in the studio?
Yes, so if you I actually have a question on this slide. So I'm wondering in your model issues when the biofilm is growing and you are trying to evaluate the effects on the microscopic permeability. So how do you ensure like continuity in the biofilm growth across your different blocks or pores? Do you have any like modeling approaches to do that, or how? I'm not sure, I didn't catch that properly.
You mean how I calculate each local pore during different time point, right?
No, so I mean at the interface of your blocks, how do you ensure that the biomass growth is continuous? So the contribution coming from each blocks is the same, basically.
Sorry, I didn't get the question.
So when you treat your porous media as like blocks to evaluate the effects of biofilm growth on your overall permeability, how do you ensure that your growth at the interface between these blocks is continuous? So do you have? I'm not sure, I didn't catch that properly. So how do you model the biofilm growth in between blocks?
Okay, it's like that. First, actually, this is based on the experimental images. We analyze that, then we can find. First, we plot all the MACs, all the pores. Then, in the different time point, we calculate how much biofilm inside, based on the total intensity of each MAC. So that means at each time point, we can calculate how much biofilm inside. Then based on my first equation, we can transfer how much biofilm inside into how the thickness of the biofilm in a pipe. You see, if you see the D2 is the thickness of the biofilm.
All right. Okay. So you use these parameters from your imaging and experiments. Okay. This is what I didn't. Okay, good. Thank you.
Okay, so we have a question from the audience coming up. So here it is. It's actually split in two parts. But first of all, he thanks for your speech, Leo. Thank you so much. And the question is, as the biofilm grows in your reactor model, the flow rate decreases, and based on Poisson law, the head loss changes. Let me put the second part of the question on, which is this one. So which should affect the delta P you are trying to keep constant? How do you confirm? So basically the question is, how do you confirm that the delta P remains constant across your model?
You mean in my second case, right?
Yeah, I think he means, yeah, because he's talking, the first part of the question was mentioning the biofilm growth. So yeah, in the second case.
Yes, actually in my second case, we develop a feedback loop. In this case, we use the pressure controller. Then we can at each time point, we can calculate about the pressure loss during the tubing and the pressure loss between two reservoirs. Then we reset a value of the pressure we want to inject.
Great. Thanks. Do we have other questions from the studio? No, this doesn't seem the case. So if not, oh, we have a question for Rima. Yes,
I
Rima,
have
go
a
ahead.
small question. I'm having a doubt about the flow rate. Like, how is the flow rate changing at the inlet and the outlet of this pipe? Like, how are you monitoring the flow rate? Are you having any idea of the flow rate, how it is changing from the inlet and exiting from the outlet of the pipe?
Actually, in this case, since we inject a constant pressure drop across the system, so actually the file of the flow rate decrease exactly goes the same trend with the permeability decrease.
Okay.
Okay, great. I think it's time to move on with the second speaker. So,
Yep. Thank you, Kira. So it's my pleasure to introduce you our second speaker, Julia Barkelew. She is a National Science Foundation Graduate Research Fellow, and she has she's a combined master student, PhD student in the Environmental and Water Resources Engineering program at the University of Texas Austin. So our research investigates how microbial metabolism and geochemical conditions interact and to control hydrogen fate and transformation in geological reservoirs. She focused on hydrogenotrophic microbial interactions among sulfate reducer, methanogens, and acetogens, and their collective impact on hydrogen consumption and storage stability. She integrates laboratory experiments with geochemical modeling and to predict how microbial activity shapes the efficiency and long-term performance of subsurface hydrogen storage system. And today our talk will focus on the hydrogenotrophic microbial dynamics in subsurface Halite reservoir. So Julia, thank you very much for being with us today, and the floor is yours.
Perfect. Thank you so much for the introduction, and good morning, y'all, or good afternoon, wherever you're located. I'm very excited to be here. I am in my second year of the combined master's PhD program here at UT. And so I wanted to share a little bit of what I've been working on for the past year or so. So to kind of get the presentation started, I wanted to start with the potential of hydrogen. And so there seems to be this really big interest in it because of its capability to be used as a clean carbon-free fuel source. It's a really big component of our energy transition and hopefully being able to kind of move away from our current dependence on carbon-based energy sources in general. And something that's really exciting for me and my research is this map that the US Geological Survey has just recently released, where they looked at regions that have necessary hydrogen sources, such as maybe radiolysis or serpentinization, reservoir rocks, and then seals to trap the gas so that hydrogen can accumulate. And then based on all of those parameters, they were able to assign a relative prospectivity value from 0 to 1. And so if it's present in sufficient quantities and recoverable, which is a big if right now, it could potentially be extracted and used directly for energy production. And so something else I just kind of wanted to point out on the map is the really nice geographic overlap with salt caverns and the really high prospectivity values associated with some of these naturally occurring geologic hydrogen hotspots. So even though this map doesn't really make any predictions on where the hydrogen is located within the subsurface, it's really promising that many of the areas with a greater hydrogen accumulation potential overlap pretty well with these caverns that have the capacity to be used for large-scale subsurface gas storage. And so in particular, especially for me and the University of Texas, it's right in our home state, the Salado Formation is of interest for us. There are currently some pilot plants that are being used for radioactive waste storage underground at WIP. And then, so just kind of putting this all together, it kind of raises the possibility that maybe naturally occurring hydrogen and engineered subsurface storage systems could maybe coexist if we really do have this big energy reserve right beneath our feet. However, like I said, the amount of extractable hydrogen will vary significantly based on the geologic formation in itself. And so if this hydrogen does exist at recoverable quantities, the amount of it that would be able to be extracted would be really, really low, likely in the micromolar concentration ranges. And so, like I had pointed out previously, the Salado Formation is kind of the area that I'm interested in, and I just wanted to show a generalized cross-section of it. And so the Salado Formation is a bedded salt cavern, and so it consists of alternating layers of relatively impermeable halite and then more permeable evaporites such as gypsum and anhydrite with carbonate nodules throughout. And so this just goes to show that in a perfect world, this would be a little bit more homogeneous and make it easier for hydrogen storage and extractability. But unfortunately, that's not the case, but it keeps our lives interesting as researchers. So I just wanted to point out that like these contrasts in lithology and permeability can create some heterogeneous flow paths and some localized fluid fill zones, which may kind of control where hydrogen accumulates and then how easily it can migrate throughout the subsurface. And so kind of taking a step back, some of those fluid-filled pockets that I was just recently talking about, here's a brief schematic of what maybe it could look like and what could be going on within those cavity sumps. And so you have your evaporites, and then as they dissolve, they'll release ions. And in combination with hydrogen, if there is hydrogen accumulation there, it leaves room for microbial growth and activity. And so the three groups that I'm interested in that are capable of utilizing these dissolved minerals, which are your sulfates and your carbonates, in combination with hydrogen, are your sulfate reducing bacteria, methanogens, and excitogens, as all three of these groups are capable of utilizing those. And then they're also anaerobic and halophilic and have been previously shown to be present within similar formations. And since hydrogen is such a favorable electron donor for anaerobic respiration, increased microbial activity in these zones can quickly draw down any available hydrogen if it's present within these cavities. And so as a result, microbial activity and then hydrogen consumption limits the amount that can be accumulated and then reduces the amount that may remain recoverable. So being able to understand how it's generated, trapped, and then most importantly for my research, biologically consumed is really important for being able to estimate how much natural geologic hydrogen we could viably extract. So just because we have sulfate ions and maybe some hydrogen located within these cavity sumps, it doesn't necessarily mean that the microbial taxa that are present are capable of utilizing them and that those reactions can actually proceed. And so since in these natural systems, hydrogen accumulates at really, really low concentrations, some of the reactions may be chemically possible. All the chemicals are there that you would need for it to take place, but they may not necessarily be thermodynamically feasible. And so there may not be enough activation energy to actually get those reactions going. And it may not be something that we should be as concerned about as other areas in the formation. And so what I did for my experiment is I used the Nernst equation as kind of an initial estimate. So I used some electrode potential data for those three microbial groups, our sulfate reducers, our acetogens, our methanogens. And then I was able to adjust it using the equilibrium constant, your Q, or I guess the Q shows how far away from equilibrium it is. And so then you can plug in your dissolved sulfates and carbonates. You can adjust for your system pH and then also your ionic strength of your brine. And so then I kind of took that and I was trying to think about what I can do with it and if there's a way that I can utilize that to kind of develop my hypothesis. And so I had a little bit of my own adaptation of a Purvey style diagram. So instead of varying pH with potential, I kept pH constant, and then I varied hydrogen activity to see if you can identify like an initial threshold for each pathway. And that's kind of what I used as an initial hypothesis in my experiments. So to kind of get that going, my overall objective was to design a lab setup where I vary the initial amounts of hydrogen that are available while keeping the other substrate concentration. So in this case, it's our sulfate and our carbonate constant to identify the minimum partial pressure of hydrogen at which we see observable activity initiate. So the first hypothesis is, in a perfect world, it's thermodynamically controlled. Your electrode potentials or your free energy diagrams, they explain everything. And so that was one of my first hypotheses, and that hydrogen consumption kind of follows those expectations. However, in a mixed culture system, there are a lot of other things at play, and a lot of microbial dynamics and interactions and competition amongst each other can kind of shift those potentials away from your pure isolated systems that are sometimes used for those initial calculations, like what I had done. And so some other studies have been able to describe these deviations and interpret them using different frameworks. And so some have been able to attribute the deviation from by kinetic competition for shared substrates, where organisms with lower hydrogen thresholds are able to draw down the aqueous hydrogen concentration that suppresses other pathways. And then others have kind of drawn a bigger emphasis on metabolic strategies, like there's some sort of energy saving complex or pathway that allows these organisms to persist under really low energy conditions, even if they maybe shouldn't be able to. So to get my experimental setup going, what I had done is I had taken this paper that has the major ion compositions of Permian seawater and used that to describe my ion composition for my brine. I'd chosen that because the Permian Sea it refers to the extensive sea that covered parts of the continents, specifically Texas, about 300 million years ago. And it matches the ionic strength of where I took my microbes from. So we took some sea sediment from the Gulf of Mexico. We chose that area because it's close by, and it consists of anaerobic halophilic microbes that already can coexist amongst each other. And so it made it a little easier than starting with pure culture, for example, and then trying to get the three groups to like each other and then stock them. And so then we ran some 16S sequencing, and I'm just showing this bar chart to show that based on their taxonomic rank of order, we were able to make sure that we had our acetogens, methanogens, and sulfate reducers within our sediment. So then after that, I dissolved the salts, and then I sterilized it, and then I moved it into the glove box to get rid of any sort of dissolved oxygen that was contained within the brine. And then for the microbial inoculum, what I had done is taken a little bit of that sediment and then suspended it into the anoxic brine. And then I combined all this together, and then I added in my sulfate and my bicarbonate, and then crimp sealed that shut. And then I took those bottles out of the glove box, and then I replaced the headspace with the adequate amounts of hydrogen that I was wanting to test. And then I let them shake and incubate. And so here is just a schematic of the reactions I was trying to track. And we were able to analyze our carbonate system and then total sulfide, hydrogen and methane using a GC, and then our aqueous analysis for our sulfate and our acetate, we were able to measure using IC. And then pH, we were able to measure by just taking an aqueous sample and then using a little pH stick. So here are the initial results from the coastal sediment experiment. So we had initially used that sea sediment, and then for the sake of brevity, I'm just going to show our results from the 1% partial pressure bottles and then the 50% partial pressure bottles. And as you can see, the initial substrate concentrations are very, very low. And so we're using 0.9 millimolar sulfate and then 6.8 millimolar bicarbonate, which was enough to set the pH and give a little bit of a buffer, and then also allow some of that to speciate into CO2 as well for the microbes to use. And so what we can see from our initial results is that in both cases, they're very energy starved. This is a very, just energy starved, which is kind of what we were wanting to test. But by looking at the results, you can kind of see that the electrode potentials that we had calculated, we just kind of hypothesized that at 1% partial pressure, there shouldn't be enough reducing power for any of the reactions to proceed, whereas at the 50% partial pressure, we should see activity from all three groups of interest. And so to kind of get that started off for our sulfate reduction, even though we hadn't anticipated to see any, we got a little bit of sulfate reduction. Then around day 20 of the experiment, it seemed to level off, whereas in the 50% bottles, it seemed to have a continuous decline. For our acetogens, we did see acetate production in both. In the 50% bottle, it's a little bit more steady, whereas in the 1% bottle, it seems to level off except for one really large spike around day 35. However, since we are working directly from that sediment and not a pure culture system, it's more likely that this could be fermentation products rather than homoacetogenic activity. And then for our methanogens, we saw no activity at the really low partial pressures. And then we saw a little bit of an uptick around day 55 for them. And so other papers have theorized that there is some sort of kinetic competition amongst methanogens and sulfate reducers. And so in our first bottle, the sulfate never dropped low enough for methanogens to be able to outcompete them for hydrogen. And then the hydrogen ran out. Whereas in the other bottle, there was still some hydrogen left in the bottle. And then once the sulfate dropped to about 0.2 millimoles, that's when we started seeing some methanogenic activity. So overall, from these experiments, what we were kind of able to see is that maybe for our specific experimental setup, there seems to be maybe our hydrogen threshold kind of lies around 0.2 millimoles, as that's when it seemed like a lot of our microbial activity kind of ceased. However, like I had said, since we're working directly from that sediment, by doing a quick mole balance for our three groups of interest, we should have 4 moles consumed of hydrogen will form 1 mole of product if they really are hydrogenotrophic microbes and not utilizing something else. And so as you can see from our mole balance, these are the results from some other bottles that we did. And then the pink ones are from what I just previously shown, that our mole balance isn't ideal, which is totally fine. We were kind of expecting that, but it was good to see that with my initial experimental setup that we were still able to track them over time and get some transient results. And so taking that information, we had done some isolation resuspension cycles. And luckily for that, we were able to decrease the amount of time that it took for us to see activity from all three groups. So for example, from our raw sediment, it took about 55 days. So they were very, very slow growers. Their biomass concentration was very, very low, and we were able to kind of isolate them, get rid of microbial groups that maybe can't grow on hydrogen, and also make our inoculum take much less time to start getting activity from. So then once we were able to do that, we moved into some different storage methods. And so the first one that we tried were glycerol stalks. And so these were made with 15% glycerol. We tried a couple different concentrations. We made them anaerobically, and then we flash froze them in liquid nitrogen and then put them in the -80 freezer. However, from the stalks at all different stock concentrations, we only saw activity from sulfate reducers. And so then we kind of tried something else. So we moved on to a Hungate tube method. So we just took a little bit of our resuspension culture with a pressurized headspace, and then we put them in the normal freezer. And luckily we were able to see all groups survived, which was very exciting. So kind of what I've learned from this in the past year that I've been working on this and as a graduate student is that I've been able to successfully grow the microbes, develop my batch system methods, and then I now have a much better understanding of the long-term storage methods for anaerobic cultures. They do take a lot of time, so I think my patience has grown as well while working on this, and I'm really excited for future work, I'll be redoing the batch reactors, which will be our time-dependent ones, but I'll be using our standardized Hungate tube inocula and then running them at our varied partial pressures of hydrogen with our 2 concentrations of sulfate. And then something that we just recently started working on maybe a month ago, so it's still very much in its infancy. So enjoy my very, very simplified schematic of our anaerobic chemostat, but we're hoping to get a chemostat up and running as well so that we can compare transient versus steady state results. So yeah, thank you very much for listening. I was really excited to present to you all today. Big shout out to both of my research groups back here at Austin. I'm happy to take any questions, or if anyone has feedback or suggestions for my experimental setup or kind of where I'm wanting to take my research, I'm more than happy to hear those as well. I'm always open to that. Thank you.
Thank you very much, Julia, for the very engaging presentation. I encourage the audience to post a question in the chat, and in the meantime, we'll start with questions from here, from the studio. So I had to have a curiosity because I'm not working with microbes and never experimental data. So you mentioned that you're using a complex mixed of different microbial groups in your experiments. So my naive question is, when you have competitive behavior, how do you ensure that this is what drives the hydrogen reduce rate of recovery? Did you run any kind of reference benchmark using pure microbial groups to check for these competitive behavior effects? So.
Yeah, great question. That's something that we had kind of gone back and forth on, trying to do some pure culture studies versus using the mixed culture. Since I'm relatively new to the project, the mixed culture was a little bit more robust and easier to work with. And so through the isolation resuspension cycles, our aim was to kind of get rid of the microbial groups. Like, for example, methanogens may use methane or other hydrocarbons available. I don't know if that's very necessary for the sea sediment that I was working with, but we were hoping through the isolation resuspension cycles to get the 3 groups of interest much larger. So there's another student working on this as well, and she's had pretty good luck with that, just isolating the sulfate reducers. But I think we are also interested in doing some pure culture tests as well, just to make sure that the competition and coexistence that we're observing isn't just because of where we pulled our sediment from, but is actually from the microbial groups that we're interested in.
Okay, cool. That's great. And then, so I was curious about like the uncertainties on your experimental results on slide 12, I guess. So you've got like error bars on your hydrogen production. You were analyzing like,
on slide 14?
12.
Okay, yes.
And you were analyzing like a rip bump at some point, explaining the sulfate behavior. But for me, the uncertainties range, like, yeah, shows like, especially on the left one at 1% pH 2.
Yes.
So the uncertainties and error bars are actually of the size of the bump you were talking about. So.
Yes. So with the really low hydrogen partial pressure, some of the problems that we've been having is, since hydrogen is so small and mobile, sampling becomes very difficult, especially at really low concentrations. You kind of start entering the zone of, is it the noise from the machine that you're running? Is it some sort of other interference? Because at those low of concentrations, what we got as a value could very well just be an error bar on the 50% partial pressures, for example. And so the way that I had done the error bars is I had just used, these were done in biological duplicates. And so I had just averaged them together and then taken the error from that. And I had assumed that it would be possible for it to be plus or minus that. But I think in the future I need to maybe fix that. I don't know if that answered your question at all.
Yes, it does. Yeah. It does.
Okay.
Thank
Perfect.
you.
Yeah. Apologies. This one is a little. There's a lot going on without the animations.
No, that's right. Do you have any more questions from the studio?
Yeah, I have a quick one actually. Thank you, Julia, for this presentation. It was
Thank
really
you.
great. So as for Sarah, I'm not into microbiology, but I liked it very much. It was really engaging. So my question is just a curiosity, actually. I mean, are you running these experiments at ambient temperature at the moment, or is it also like temperature controlled?
Yes, so we're running them at ambient temperature. So the shaking incubator that we have is open to the atmosphere. And since the microbial groups that we got come from Texas, which has a very neutral temperature almost year round, that's kind of how we had decided to do that.
Okay.
But
Like if you, I mean, as you are doing all of these, right, to sort of mimic what happens even in the deep underground, I guess.
yes,
Do you
it'll
think-
be much hotter. So
but
yeah,
so-
I think temperature dependence. Sorry, I didn't mean to cut you off.
No, don't worry. Go ahead.
Oh yeah, definitely. So in the subsurface, the temperature dependence will be much greater, and also these are being run at atmospheric pressure, and so that's another thing that we've discussed. It just becomes a little bit more complex trying to run these at higher temperatures than ambient, since we have to sample, and so like for our gas sampling, we have to do it manually, and so you just have to be a little bit quick with it. But that is definitely something that we're interested in looking at, because I think it'll change the results, and it would be very interesting to see.
Great.
Yeah, I think we have like a follow-up question on that, on the pressure from someone in the audience. So you were asking if you intend to study at the behavior under storage condition and elevated pressure.
Yes. Yeah. That's something we are interested in doing. But I think to just get the methods defined and then also get our culture defined, it was easiest to work with atmospheric pressure. But I know that both of my advisors are very interested in looking at the temperature and pressure dependence of these cultures, since that'll better mimic what would actually be happening within the subsurface.
Yeah, that sounds promising, like perspective for the future years of PhD. Any more questions from the studio? Okay, so I guess no, we don't have any more questions from the audience either. So I think we can conclude here. Thank you again for the both of speakers. It was really interesting presentations. And we would like to advertise our session for next month, scheduled on the 17th of February. So it will be a Korean special with a slightly different time. So for our European audience, please note that this is 2 a.m. Central European time. So if you may need a very large cup of tea or coffee if you want to join live. So it's totally up to you, but otherwise, please be aware that you can still watch the recorded version on YouTube afterward. So our speakers would be Wenqiao Jiao from Yonsei University and Taeyong Kim from the Korean Institute of Science and Technology. So please attend the session if you can and share the word about the YouTube video. And yeah, so but again, if you would like to be involved as speakers or want to subscribe to a mailing list, you can contact us at porous@gmail.com. Feel free to reach if you have any suggestions on how to improve the sessions as well. Thank you, everyone, and stay tuned for the next PMTTT. Bye.